Dr. Josh Leeman is Vice President of Business Development, Search and Evaluation at uniQure, where he leads establishing new gene therapy business opportunities. Please welcome Josh and his talk, Focal Administration of AAV Therapeutics for Severe Neurological Diseases.
I wanted to thank Tyson and Eric and the Dino team first. I think it's wonderful that you've put this together. There's rarely a chance where the community, a specific community like this, can get together, share ideas, experiences. And so it was a lot of fun listening to these guys, listening to Jim talk about his 40 years of experience and the rack and everything else. At the beginning of the day, Eric walked us through some slides. And one of those slides showed a spectrum of patient need and how that leads to this genetic advocacy and the agency. Today I'm going to speak about two examples that we're working on that fall on different parts of that spectrum. The first one is Huntington's disease. This is AMT-130. You may have heard about this in the last few weeks and months. Huntington's disease affects approximately 70,000 people in the United States and EU. It's absolutely terrible. Patients start off with mobility disorders, chorea, neuropsychiatric and cognitive deficits, always leading to death. At uniQure, we're employing AAV5 like we did with Hemgenix, and we are delivering a microRNA directed at Huntington's gene. Once in the cell, the microRNA knocks down both full-length wild type and mutant Huntingtons, as well as this toxic aberrant splice variant called, that includes exon 1. HTT exon 1 is the name of the splice variant. And so that was sort of a lucky thing when we designed the microRNA that also knocked down that variant. So we believe AMT-130 has potentially first-in-class, best-in-class, and that's really because of some of these advantages. So the title of the talk was about focal delivery. I know Dino is working on next-generation capsids that hopefully can get us past that, but right now, this one-time administered gene therapy is done through a surgery directly to the caudate and putamen, but it's this precise focal delivery that we think drives the biodistribution and target engagement in the critical areas. It also minimizes systemic exposure, which we all understand the limitations there. And so while this is a surgery, it really is something that a lot of physicians have experience with, and we believe it can be broadly performed. So just a very high-level touch point on what the data looked like from our phase 1/2 clinical trial. Here we're looking at two clinical endpoints. At the top is the CU-HDRS scale. That's a composite scale, on the bottom is TFC, total functional capacity. In the gray bars are what the natural history shows happens to patients over a 36-month period. In orange are what we observed in our treatment groups. And this is high dose. So at high dose, we saw a significant reduction in disease progression by 75% as measured by CU-HDRS and 60% based off of TFC. So here's a status update. So like I said, we saw statistically significant slowing of disease progression by both of the clinical endpoints. One thing I didn't show is that while there is an initial spike in NFL due to the surgical procedure, over the following year, this rapidly reduces and it falls below baseline as compared to natural history, actually. It's generally well tolerated with no SAEs related to the drug. And now to the regulatory question. So 2 weeks ago, last Monday, you guys might have seen a press release we had following up on our pre-BLA meeting with the FDA. Essentially, what we thought we had agreed upon was not the feedback we got from the FDA. So we have conviction for these patients. We believe in the data we have generated. And so over the next month, we'll anticipate release of the minutes from that meeting and plan to work with the FDA in any way we can. Fortunately, that's all I can say about that right now. It's rapidly evolving, and we're trying to understand it better. All right, so the second story I'd like to share is AMT-260. This is for refractory mesial temporal lobe epilepsy. And so these patients have actually a number of therapeutic options. And so to the point of this meeting, we believe that this provides them a different path, so they choose. So refractory means that they have lost or they do not receive any benefit from the drugs available, but they do have the the opportunity for hippocampal resectioning or a lobectomy, essentially destruction of brain matter. So not unsurprisingly, patients tend to go a very, very long time if they so choose to go that path to make that decision. So I think maybe 2% of patients who are eligible undergo those procedures on an average of 15 years. So they sit on this, decision for a very, very long time. So what we're doing here is, and this is described in the cartoon at the bottom, but in normal neurons, kainate receptors modulate neurotransmission, neuroplasticity, and network excitability. In TLE, they've been implicated in the disease state. So these receptors are made up of subunits, GLUC1 through 5, which are encoded by the genes GRIC1 through 5. So AAV9 uses, AMT260 uses AAV9, which deliver 2 microRNAs, which are targeted at GRIC2 mRNA, thus reducing the expression of GLUC2. And so treatment with AMT260 leads to the reduced expression of this kainate receptors in TLE. Again, this is focally administered via stereotactic surgery to the hippocampus using one single administration, one single trajectory, and is, again, MRI-guided. So this is the data that we can share right now. It's a couple months old at this point, but the most important thing is we don't see any treatment-related SAEs. It seems pretty clean. And in the one patient we've described publicly, we see some striking results here. So this patient had reported seven seizures in a month retrospectively and 5 during the screening period. But following treatment, the number of seizures reduced to 0 with a little blip there in month 3 and has remained at 0 throughout the entire follow-up. I didn't show it here, we haven't publicly released it, but there are an additional 2 patients that we're following. So that's that. I think that, there are clearly challenges. They're different than we heard earlier, but hopefully Jim's right and that there's a lot of hope and we can get past these. Thank you, guys.